Greetings, everybody. So today we are going to discuss the one shot of our next chapter, which is bioenergetics. Bioenergetics, first and foremost, starting with the very basics, that it is a um, branch of biology. It is a branch of biology which deals with the capturing and conversion of energy from one form to another in the living system. Hence, it is known as bio, meaning life, and energetic means energy, yeah, which means that conversion of energy occurs in living system and and by its utilization in metabolic activities, which is known as bioenergetics. Now, bioenergetics includes uh, mm, several mechanisms, apart from which a major mechanism of them is the conversion of ATP into ADP and ADP into ATP. First, we better need to understand what is ATP. ATP is the common currency, energy currency of the cell, and is composed of adenine and ribose sugar with 3 phosphate energy. Each bond, each the last two phosphate which are connected through bonds which are known as high energy bonds. They give an energy of around 7.3 kilocalorie per mole upon breaking those bonds, which is a high energy. Moreover, when it comes to uh, the structure of ATP structure is in such a manner that here we are having ATP structure is in such a manner that here there is an adenine nitrogenous base. Adenine nitrogenous base. Let me just try to draw the structure. There is a ribose sugar, and that ribose sugar is having at the first carbon of this ribose sugar there is attachment of of adenine nitrogenous base. Adenine nitrogenous base. This forms the structure adenosine. And when on the fifth carbon there is attachment of phosphate, this is known as an adenosine monophosphate. Likewise, if there is another sugar, another phosphate attached, it is known as adenosine di, and it is known as adenosine triphosphate. We have previously studied. Now, coming to the main point here, it is a very important factor that whenever there is an addition of phosphate group, and whenever there is an addition of phosphate group, phosphoric acid to ATP, sorry, to a compound, if there is an addition of phosphate group or phosphoric acid to a compound, it is known as phosphorylation. And if there is removal uh, ATP, if there is removal of phosphate group, phosphoric acid, then it is known as dephosphorylation. Let's study these terms as well, as we'll be going to, discuss this in detail. Under cellular condition, ATP produces 37.3 kilocalorie or conversion of ATP into ATP and act as a mediator capable of receiving energy. Transfer the energy to drive another reaction. Now here, inorganic Pi represents phosphate which is obtained from an inorganic source, which means inorganic phosphate represents a phosphate which is obtained from an inorganic molecule, inorganic source like phosphoric acid. And the process of ATP formation from ADP and phosphate is called phosphorylation. There are three types of phosphorylation occurring in living organisms, among which first is photophosphorylation. Photophosphorylation here is is a process in which ADP is converted into ADP. ADP is converted into ATP by the addition of an inorganic phosphate through utilization of energy of sunlight. At that moment, that energy of sunlight utilized to drive the process of converting ADP into ATP by the addition of inorganic phosphate is known as photophosphorylation. It occurs in the thylakoid membrane of chloroplast and utilizes the energy of light. When it comes to oxidative photophosphorylation, photophosphorylation occurs in autotrophs. Photoautotrophs, both who perform non-classical photosynthesis as well as classical photosynthesis. We are going to discuss that as well. What is classical and non-classical photosynthesis? Oxidative phosphorylation. In this type of phosphorylation, ATP is formed by using the energy of oxidation, right? During metabolic reactions in cell. The site of reaction is cristae of mitochondria, in which ATP is formed by energy of oxidation during metabolic reaction in cell. It occurs in the third step, which is known as the electron transport chain of uh, the respiration, in which oxidative phosphorylation occur, as a result of which ATP is converted into ATP using energy of oxidation. The phosphate obtained in this case is also inorganic. Now, substrate level phosphorylation. In this type of phosphorylation, one substrate provides phosphate and energy to another substrate. Such a such a In such a manner that here we are having uh, an enzymatic reaction going on. Living organism produces ATP through two processes: photosynthesis and respiration. When it comes to photosynthesis and respiration, now within photosynthesis there's a step in which in, in photosynthesis there's a step in which 3-phosphoglycerate 3-phosphoglycerate is converted into, wait, 6 molecules of 3-phosphoglycerate are converted into 6 molecules of 1,3-bisphosphoglycerate. Now here 3-phosphoglycerate represents a structure that here glyceric acid is a structure which is basically having 3 carbon sugar is there, glyceric acid, and here a 3 carbon acid is there, sorry, and on the third carbon there is present of phosphate, so it is known as 3-phosphoglycerate. Now with the help of an enzyme which is transphosphorylase, it causes to give a compound which is known as 6 molecules of 1,3-bisphosphoglycerate 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 or diphosphoglycerate. Now in this case 1,3-bisphosphoglycerate represents that there is an addition of a phosphate group on each of the phosphoglycerate on the first carbon as well, due to which it became 1,3-bisphosphoglycerate using the enzyme transphosphorylase. Now in that case the additional phosphate group which is obtained is from the conversion of 6 ATP into 6 ADP. 6 ATP into 6 ADP plus release of 6 inorganic phosphate which is utilized in this process. So in this case, the phosphate which is obtained is from an organic source which is ATP, due to which the phosphate which would be obtained would be represented by PO, which means organic phosphate. So in substrate level phosphorylation, the phosphate which is obtained is organic phosphate. Apart from which, when it comes to some other part, um, we are going to discuss the photosynthesis. It is an oxidation reduction process in which plants manufacture carbohydrate by using carbon dioxide and water in the presence of sunlight with the help of chlorophyll. Or it can also be defined as a biochemical anabolic process during which glucose is manufactured from carbon dioxide and water in chlorophyllous cells. In chlorophyllous cells, in presence of sunlight, and oxygen is given out as a byproduct. The chemical energy obtained is ATP and NADPH2. Now, role of light comes in such a manner. Light comes on Earth in packet called quanta. A single quantum light is known as photon. A light travels in waves. Plants can use only a small portion of visible light that falls on leaves. Light is composed of 7 different colors, Bibgyor, which are known as violet, indigo, blue, green, yellow, orange, red. Their wavelengths are also shown over here, such that the wavelength of light ranges from 380 to 760 nanometer, and 380 showcasing the frequency 380 showcasing the wavelength of violet, and 760 the wavelength of red light. So here As you can see, when light falls, it rapidly transferred to the chlorophyll molecules. Uh, chlorophyll molecule present in the photosynthetic reaction center. Now here, the for absorption spectrum for chlorophyll, absorption spectrum for chlorophyll, absorption spectrum for chlorophyll shows that absorption is maximum in blue and red. Peaks are found between 430 and 430 and 600 something, 430, 430 and 630 nanometer, such that it is between 430 and 600 nanometer, is between 430 nanometer and 670 nanometer. While when it comes to 600, 430 and 670 nanometer, apart from that, in photosynthetic pigments, differential in photosynthetic pigments, differential absorption spectrum is obtained. A relative effectiveness of different wavelength color of light is driving photosynthesis, called action spectrum of photosynthesis. It is observed. It is it is over here, can be determined in such a manner that here substances in plants that absorb visible light are called pigments. Different pigments absorb light of different wavelength. These pigments capture light energy and convert it into chemical energy. The important pigments are chlorophyll, carotenoids, and phycobilins. The one which we are going to discuss over here is chlorophyll and carotenoids. Phycobilins will be discussed in the protoctist. So here chlorophyll are there are various types of chlorophyll named as A, B, C, D, and E, E and F as well. Here such that A, B are present in plants, but in algae there is also presence of these additional pigments known as C, D, E, and F. Apart from that, when it comes to the formula of chlorophyll A has its formula as C55H72O5 and 4Mg, as well as when it comes to the of structure, it is previously discussed in our videos that here it is having bluish green and C55H72O5 and 4Mg, such that it is having a methyl group, while when it comes to the chlorophyll B structure, it is C55H70O4, C55H70O6 and 4Mg, such that instead of methyl group there is an aldehyde group, due to which it appears yellow green. A photosystem consists of a cluster of few hundred molecules of chlorophyll A and B and carotenoid molecules, and chlorophyll A is present in almost every photoautotrophic cell, including cyanobacteria. Many of the cyanobacteria are also having it. But when it comes to chlorophyll B is present in all of the plants and algae except dinoflagellates and diatoms, brown algae, dinoflagellates and diatoms, as well as brown algae. And chlorophyll molecule has two main parts: one flat, one square, light absorbing hydrophilic head, and the other long hydrophobic hydrocarbon tail. The head is complex, made up of small pyrrole rings, four small pyrrole rings, four small pyrrole rings. Where is it? Pyrrole rings joined by magnesium in center with nitrogen to porphyrin rings. The structure is in such a manner there are four carbon atoms and one nitrogen atom, which attaches with the central magnesium atom and forms a porphyrin ring. Along hydrocarbon tail, which is attached to one of the pyrrole ring. There are two tails, phytol tail, which is C20H39, and another tail which is known as methyl ester, which is attached with it. Methyl ester is short, so we usually don't write it. And chlorophyll molecule is embedded in the hydrophobic core of the thyroid membrane of the cell. Chlorophyll molecules mainly absorb violet, blue, and orange red light, which is in the range of spectrum 430 to 670 nanometer. Green, yellow, and indigo are least absorbed to chlorophyll, therefore they are green. The most important pigment is chlorophyll. It is also found in abundant quantity. It is also found in abundance. It is involved in light dependent radiation, converts light energy into chemical energy. Moreover, chlorophyll B is found with along chlorophyll A in all green plants and green algae. When it comes to carotenoids, these are lipid in nature. They are basically lipid in nature. as well as they are present in the thylakoid membrane, such that they have, these are basically terpenoids. These are terpenoids, and they have, uh, isoprenoid units with isoprenoid ring at one end or both end, such that one of the most abundant form in carotenoids includes carotenes and xanthophylls. Xanthophylls basically include several examples, such as lutein is the one xanthophyll which we usually see during autumn season when the leaves are being, defoliated. Moreover, uh, when it comes to other xanthophylls, such as fucoxanthin is present in brown algae, as well as zeaxanthin present in corn, z, as well as when it comes to uh, cryptoxanthin, it is found in papaya. So these are some of the xanthophylls. When it comes to carotenes includes beta carotene, alpha carotene, gamma carotene, among which beta carotene is the most abundant, and these are yellow and red to orange in color, such that red to orange one are known as the carotenes, and yellow to orange ones are known as xanthophylls. Carotenes can absorb the wavelength of light of blue violet, can absorb the wavelength of light, can absorb the oops can absorb the wavelength of light of blue violet range efficiently. This chlorophyll cannot absorb and transfer it to chlorophyll B, and then transfer to chlorophyll A, and from here energy transfer to light reaction occurs. The order of energy transfer is shown in the below diagram. In such a manner, carotenoids transfer it to chlorophyll B, and chlorophyll B transfer it to the reaction center, which is chlorophyll A, because chlorophyll A is a major molecule which will tend to lose its electron as from its magnesium atom, as a result of which the electron being transferred to the other electron acceptor, will tend to form the a non-cyclic or cyclic photophosphorylation. Now, excessive light can damage the chlorophyll. Instead of transmitting energy, instead of transmitting energy to the chlorophyll, some carotenoids can accept energy from the chlorophyll. This is known as photoreception. Role of photosynthetic pigment in absorption conversion of light energy. Different pigments have their specific absorption spectra, therefore they absorb light of different wavelength, but all accessory pigments transfer the energy to chlorophyll A and then to the carrier of photosystem. Absorption spectrum of chlorophyll A and B. Chlorophyll A absorbs violet and orange light with a wavelength of 650 to 700 nanometer. Chlorophyll B absorbs blue and yellow light of 450 to 500 nanometers. These are some important MCQs points. After that, when it comes to arrangement is in such a manner that photosynthetic pigments are present in groups known as photosystems. When photos reaction takes place in photosystem, chlorophyll and other molecules are arranged in a systematic way in an antenna complex, uh, such that the main reaction center involves the chlorophyll A, and rest of the molecules which are arranged side by side with it are known as accessory pigments arranged in an antenna complex. This complex contains few hundred chlorophyll A, chlorophyll B, and carotenoid molecules in the form of cluster. When light is captured in the cells of leaves, it is rapidly transferred to the chlorophyll A molecules present in the photosynthetic reaction center. In this center, the first light-driven chemical reaction of photosynthesis occurs. This photosynthesis of two types. The photosystem is of two types: antenna complex and reaction center. Here, as you can see in the diagram, that reaction center is in such a manner that here light, sunlight is poured over the antenna complex, as a result of which that antenna complex, after absorbing of light, loses transfer light energy to the reaction center, transfer that light energy to the reaction center, which is a chlorophyll A, which loses its two electrons which are delivered on the primary electron acceptor. One more thing, that the reaction center is in the form of a dimer, which means there are two chlorophyll A molecules attached. So the reaction center is in the form of dimer, due to which the magnesium atom from both releases two electrons to the primary electron acceptor. And one more thing over here, that here the energy of transfer from the primary energy transfer from from the antenna complex to the reaction center is without the transfer of electrons. Such type of energy transfer is known as resonance energy transfer. Resonance energy transfer. After that, Role of water. This is a short topic which we are going to cover. Photosynthesis is a redox process. It requires proton and electron to fulfill this requirement. Water is split into ionic form with release of electron called photolysis of water. Called photolysis of water. After that, called photolysis of water. Here, this is a first step in which we know that carbon dioxide basically is involved in formation of glucose, but when it comes to the role of water, role of water is in confusion that where does the water go in the reaction, because H, the hydrogen, is utilized in formation of glucose, but oxygen can be utilized to form glucose either, or is it utilized to would form oxygen. So it was a confusion at that time. So there was a scientist named as Neil, and he basically unveiled the hypothesis which was on the basis of photolysis of water. He said that H2O is obtained from plant. H2O is obtained from it is basically involved in the the oxygen which is evolved from the plants is obtained from the oxygen present in the H2O, and this process in which the H2O is split into the ionic form with the release of electrons is known as photolysis. And here, as water molecules are split, their oxygen atoms combine to form the molecules of oxygen. So it is concluded that the water thus provides proton and electron necessary for the reduction step, leading to assimilation of carbon dioxide. It is now confirmed that the oxygen released as a byproduct is a part of water but not carbon dioxide. It was first proposed by one Neil in 1930. It was first proposed by one Neil in 1930, where he gave his hypothesis about uh, that water is the oxygen evolved from plant is obtained from the water rather than carbon dioxide. Uh, on what basis did he do that? He did that on the basis of finding purple sulfur bacteria, green purple sulfur bacteria. The green purple sulfur bacteria had a characteristic that instead of uh, you know utilizing water, it utilized hydrogen sulfide. It utilized hydrogen sulfide, and as a result of utilizing hydrogen sulfide, he noticed that the green purple sulfur bacteria, alongside utilizing hydrogen sulfide, alongside uh, utilizing hydrogen sulfide, had a byproduct released within the green purple sulfur bacteria, which was sulfur, which means it indicated, and he came to know after looking at that reaction, that instead of oxygen, there was sulfur, due to which sulfur gas was being released from green purple sulfur bacteria, as a result of which he was then determined that this is why H2O is the major source of oxygen present in as a byproduct in the process of photosynthesis. So this was his conclusion. Apart from that, when it comes to some other part, here in 1940, an experiment was conducted by some scientists on an algae known as Chlamydomonas through labeling of isotope radioisotope with the H2O and carbon dioxide as oxygen. In the first group, group 1, they basically utilize a radioisotope through which they produce a radioactive water molecule. And when they produce a radioactive water molecule and allowed it to be utilized in the process of photosynthesis, they obtained the result that the oxygen which was evolved was radioactive. Likewise, when group 2 plants were tested, group 2 plants were actually given radioactive oxygen present within the carbon dioxide, as a result of which when they provided that oxygen to that carbon dioxide having oxygen to plant, they got the result that glucose which was obtained contained radioactive oxygen isotope, which indicated that here uh, the clear evidence was obtained that H2O is a major source of oxygen evolved during the process of photosynthesis, and carbon dioxide is a source of oxygen obtained during the process of photosynthesis. Apart from which, when it comes to role of carbon dioxide, it provides a carbon for the synthesis of carbohydrate. Opening and closing of stomata have an important effect on the regulation of photosynthetic activity. Investigation shows that there is increase in photosynthesis with an increase in carbon dioxide concentration at different light intensities. Increase in carbon dioxide concentration at different light intensities. the process in which carbon dioxide and water combine in the presence of sunlight in chloroplast, producing product glucose. Water and oxygen as a byproduct is known as photosynthesis, in which there is a reduction of in which there is a reduction of glucose, in which there is reduction of carbon dioxide, as well as there is oxidation of water, reduction of carbon dioxide, oxidation of water in order to produce glucose. Apart from which, when it comes to the other part, here the reaction is simply written down on your screens. And here, apart from that, when it comes to other part, here we are having the reactant and products of photosynthesis. Reactant and products of photosynthesis are in such a manner that here the reactants of photosynthesis are water, carbon dioxide. Reactant of photosynthesis are water, carbon dioxide, high energy. Reactants are basically the carbon dioxide, water, and light energy in the chlorophyll, and products are the glucose, water, and oxygen. And now we are going to discuss about how the process occurs. First step into light reaction, previously known as, you know, light reaction, also known as the light reaction. It is also known as light dependent reaction, or in other words, we also call it light dependent reaction, or in other words, we also call it as light reaction. It is basically also known as um the it is also a process which is known as, you know, light dependent reaction, as well as it is also known as photophosphorylation, in which cyclic and non-cyclic photophosphorylation occurs. Now here, and this process was also discovered by a person. uh, Here, so in the light dependent reaction, chlorophyll and other molecules are present in the membrane of thylakoid and carry light energy and convert some of it into the energy carrier molecules ATP and NADPH to their energy will be consumed to produce carbohydrate. Here, in such a manner that, as you can see, steps of light reaction in such a manner that here we are having arrangement of uh, arrangement of thylakoid arrangement of photosystem, the thylakoid membrane. There are two photosystems involved: photosystem 1, photosystem 2. When it comes to photosystem 1 is associated. Photosystem 1 is associated with uh, cyclic photophosphorylation. Photosystem 2 is associated with non-cyclic photophosphorylation. Photosystem 2 is associated with evolution of oxygen as well as photolysis of water. Photosystem 1 is not associated and is only associated with the production of ATP. So let's see how does it make sense. So here, as you can see, first part includes electron transport chain. Electron transfer chain occurs such a manner in chloroplast. Electron transfer chain is composed of three parts: chlorophyll molecule, membrane bound proteins, and electron carriers. First includes membrane containing the following types of four groups of complexes are there: photosystem 1, photosystem 2, cytochrome Bf complex, and ATPase complex. Cytochrome Bf complex, it is known as a complex even though it it also has a dual role in it, such that it acts as protein, membrane bound protein. It acts the carrier, such that it transfers electron uh, from the stroma to the thylakoid space, as well as it acts as a complex to pump protons from the stroma to the thylakoid space, as a result of which it acts as a complex, and it is a BF complex, which means it is having two components: B component and F component. ATPase complex. Electron carriers transport the excited electron between complexes. The name of mobile electron carriers is plastoquinone, plastocyanine, and ferredoxin. Plastoquinone as well as plastocyanine, copper containing pigment, ferritoxin, iron containing pigment. After that, both photosystem 1 2 contain chlorophyll A molecule in center. The chlorophylls are alike to all other chlorophyll A molecule. Photosystem 1 chlorophyll A. Photosystem 1 chlorophyll A has chlorophyll A has maximum absorption at 700 nanometer, while in photosystem 2, the maximum absorption occurs at 618 nanometer. The difference only comes because of the proteins which are associated. Even though both of them are having the same chlorophyll, same number of same accessory pigments, the difference only comes with their protein molecules. The proteins which are attached with them cause to decrease their wavelength of light, which is absorbed by them. So here, this is why we also represented them as P700 and P680, such that photosystem 2 absorbs 680 nanometer, photosystem 1 absorbs 700 nanometer. Photosystem 1 is also known as pigment system of 1, and photosystem 1 basically it is uh, photosystem 2 is an uplifted region such that it is present on the upper region. It is present on the granal side, and photosystem 2 is present on the stromal side. Photosystem 1 is present on the stromal side, photosystem 2 is present on the granal side. So in such a manner, here there is a granum, here there is a thylakoid, and photosystem 2 is present over here, photosystem 1 is present over here. Photosystem 2 granal side, photosystem 1 stromal side. So this is how the structure is formed. Now steps of electron transport is in such a manner. Electrons of chlorophyll combining with compounds in pigment system to produce ATP. An energy rich compound is produced. Such steps, the steps are as follows. When light energy is absorbed by the antenna complex or the accessory pigments of the photosystem 2, is this absorption of light energy causes the accessory pigments to transfer that light energy towards the reaction center, which is chlorophyll A, in the form of a dimer, as a result of which that tends to absorb the light energy, and when it absorbs that energy, its electron gets excited and it releases its electron from the magnesium atom, not another atom, magnesium atom, because it is a center atom. So two electrons are released because the photosynthesis here it is in the dimer form. So two electrons are released, and chlorophyll of pigment system 2, it absorbs sunlight and excited electrons. It absorbs electron and excited electron. First captured by primary electron acceptor, which is pheophytene, and from pheophytene the electron is then transferred to the plastoquinone. When plastoquinone not only takes the plastoquinone here is a compound which not only takes the electron, but alongside electron it also accepts the proton from the stroma, as a result of which it becomes reduced, and as a result of reduction occurring within it, it would be now known as plastoquinol, due to which it would be represented as PQH, plastoquinol. Now the P680 produced by this primary charge separation and electron transport to re-reduce by electron from water, such that photolysis of water will occur. Now photolysis of water occurs in photosystem 2 because of a special complex present in photosystem 2, uh, which is known as manganese cluster Z complex. Manganese cluster Z complex present over here, which basically is associated with photolysis of water, such that manganese cluster Z complex causes a photolysis of water within the thylakoid space, as a result of which the process causes to evolve oxygen. The process causes to evolve one oxygen molecule and four protons by the splitting of two water molecules, as a result of which four water molecules and one molecule of oxygen into the lumen is released, and the excited electrons are transferred from the primary electron acceptor to the plastoquinone, and the chlorophyll structure is it becomes re-reduced by the electron, and after that, when it comes to the other part, this is thought to be the rate limiting step of the electron transport, which is the transfer of electron from the plastoquinol to the cytochrome BF complex, because it is a rate limiting step, because in this case, when electron is transferred from the plastoquinol to the cytochrome BF complex, this step tells us whether there would be a further requirement of ATP during the process of light reaction. And if there is a further requirement of ATP during the process of light reaction, then instead of performing non-cyclic photophosphorylation. The process which would be occurring over here would be cyclic photophosphorylation in order to yield more of the ATP, because in cyclic photophosphorylation there is only production of ATP and nothing else, as well as in non-cyclic photophosphorylation, which we are studying right now, as yield of ATP as well as NADH, pH, and oxygen. So this step is a rate limiting step which tells us whether to perform non-cyclic or cyclic photophosphorylation. Now the phospho now here the plastoquinone releases the protons into the lumen. Plastoquinone releases the protons into the lumen. Where is it? Plastoquinone releases the protons into the lumen. When the electron transfer to plastocyanin is reduced, which is present from in cavity, such that plastoquinone transfers a proton as well as electron to the cytochrome BF complex, which pumps the proton into the thylakoid space, while the electron is then pumped from the cytochrome BF complex to the plastocyanin, due to which the plastocyanin becomes reduced, as a result of which plastocyanin now transfers this electron towards chlorophyll A naught. It transfers this electron towards the, plastocyanin transfers the electron towards the photosystem one, and from the photosystem one, when the photosystem one accepts the electron, the photosystem one, after accepting the electron, causes to release excite its now here when there's something which is going to happen over here, which is that a plastocyanin, when before accepting of electron by the uh, photosystem one from the plastocyanin, the chloroph the photosystem one will tend to lose its electron. Photosystem one will tend to lose its electron, such that it will also have its accessory pigment excitation, due to which there will be resonance energy transfer, due to which the electrons will be transferred towards the reaction center. Reaction center will lose its electron, as a result of which it will become oxidized. Now it would be re-reduced. It was previously in reduced form, then it became oxidized, and then it would be re-reduced by the plastocyanin's additional electron which is present with it. So in this case, the additional electron of plastocyanin will be given to it, as a result of which it will be re-reduced, and the electron which is lost by the photosystem 1 will be basically now accepted by the primary electron acceptor of the photosystem 1, which is known as chlorophyll A naught, which is a modified chlorophyll A naught, which is primary electron acceptor of photosystem 1. It also absorbs sunlight from the chlorophyll A. The electron is transferred to the ferredoxin. Chlorophyll A naught, the electron is transferred to the ferredoxin, which is iron containing, and to ferredoxin reducing substrate. From ferredoxin reducing substrate, the electron is transferred to the ferredoxin, an iron containing protein and enzyme called NADP reductase, then transfers the electron from the ferredoxin to NADP. This NADP with two electrons receives a proton from the stroma and reduces the NADP into NADPH2. Now at that moment NADPH2 is in now in reduced form, due to which it stores high energy, which is used in the preparation of simple carbohydrate during dark reaction. So now NADPH2 is considered as a reducing power. It is considered as a reducing power because it has proton with it. It has reducing power, whereas ATP is considered as the assimilating power because it has the power to absorb, power to make something absorb. So in this case, it is assimilating power and reducing power. It stores high energy, which is used in the preparation of simple carbohydrate during dark reaction. Moreover, when it comes to some other part here, uh, as you can see, this is a diagram in which it is diagrammatically informed, but when it comes to the process which is necessary over here, formation of ATP. One more thing, that here there will be requirement of formation of 12 NADPH2 molecules and 18 ATP molecules. uh, so in order to obtain this, three proton pumped three proton pump will give one ATP molecule, three proton give one ATP molecule, two proton give pumping of 3 proton give 1 ATP molecule, and addition of 2 proton gives 1 NADPH2 molecule. Now we need 12 NADPH2, which means 24 proton required for 12 NADPH2, 18 ATP molecules, which means 3 into 18, is equals to 56, due to which 56 proton are required here. 56 plus 24 around, sorry, it's 3 18s, 24, it's 54 electrons. 54 electrons, 54 plus 24, it is around 60, 78. So 78 protons are required in this case during the electron transport process. Now let's see how, 78 protons are required. When the electrons moves through the chain, its energy is used by thylakoid membrane to produce ATP. Now since the protons are being pumped into the thylakoid space, there is the concentration of proton in the thylakoid space increase, the concentration of proton in the stroma decrease, due to which there's production of a gradient, which means gradient is produced of more of the positive charge in the thylakoid space, less of the negative positive charge in the stroma, due to which electrochemical gradient is produced, due to which positive charge in the thylakoid space, negative charge, uh, less positive charge in the stroma, due to which this gradient causes to pump the protons from higher potential to lower potential, such that protons now will tend to be pumped from the higher potential to lower potential, I from the positive region to the negative region, such that positive region is what? Positive region is the thylakoid space, negative region is stroma, due to which when it will be pumped from electrochemical gradient through a special complex which is known as ATPase complex. ATPase complex. That ATPase complex will tend to pump those protons. It will the protons, due to the formation of electrochemical gradient or chemiosmotic gradient, will tend to move from the higher concentration gradient to lower concentration gradient, due to which uh, they will tend to generate a force through the ATPase complex, because they'll be moving through the ATPase complex, due to which when they'll be moving, they'll cause to produce a force through the ATPase complex, which is known as the protomotive force. Protomotive force, which will tend to give energy to the protomotive energy to the ATPase complex, and utilizing that protomotive force, that ATPase complex will now uh, become activated, such that as we are having F naught F1 particle, we are having CF naught CF1 particles over here, due to which CF naught CF1 particles are in such a manner that CF naught and CF1 is having similar structure to ATPase complex present in mitochondria. CF naught, let me just try to draw it. Let me see if there's a diagram available. Yes, there's a diagram available. CF naught CF1 particles. CF naught CF1 particles, it's not here, so I'm just going to draw it. So here, as you can see, CF naught, CF1 particles are there. CF naught particles basically contain, these are CF naught and CF1. CF naught particle, basically they contain an enzyme which is known as ATP synthetase enzyme. Whereas CF1 particles, CF naught particle basically contains ATP synthetase enzyme, and these are basically transmembrane protein which are present here. Transmembrane protein which are present over here. Transmembrane protein present over here. So CF naught particle is a transmembrane protein which is present, having the ATP synthetase enzyme, which causes to act as ATP synthetase enzyme and convert ADP into ATP, ADP into ATP, ADP into ATP. This is the first process. Secondly, F1 particle, basically it is a particle which is a bit protruded outside the stroma, protruded outside the stroma. So this is how it goes, and here, as you can see, uh, this is how the process will occur. And when the protomotive force will be utilized by the ATPase complex, it will tend to utilize that force in order to convert ATP into ATP, using that protomotive force, as a result of which there will be production of ATP molecules. And this type of production of ATP molecules through utilizing the energy of sunlight will be known as photophosphorylation. And what type of photophosphorylation would that be? That would be a non-cyclic photophosphorylation. And this process in which it is produced is known as chemiosmosis or chemiosmotic synthesis. Now here the term osmosis might confuse you, that why osmosis is here. So osmosis, the process which is occurring over here is not actually of osmosis, because in osmosis we are having the movement of water molecules from higher concentration to lower, to from its region of higher concentration to its region of lower concentration through a partially permeable membrane. But here there is no involvement of water molecules. Instead, there is involvement of protons. So proton basically are what? Protons are part of water molecules. So these protons being pumped from higher concentration to lower concentration through a partially permeable membrane resemble with the process of osmosis. But in actual, these are not, osmosis is not occurring over here, but this process resembles with the process of osmosis, due to which it is known as chemiosmosis, because it is occurring uh, a process which is similar to osmosis due to electrochemical gradient formed over here. The process which is occurring over here would be the diffusion. And what type of diffusion would be there? It would be facilitated diffusion. And once we are done with it, here we are good to go with it. And now when it comes to the second part, um light independent reaction. Before that, there's a simple topic which I would like to highlight there, the which is about the cyclic photophosphorylation, such that, as you can see, this is the photophosphorylation occurring. There is also cyclic photophosphorylation. Cyclic photophosphorylation is simple. It is the process in which there's no photolysis occurring, no oxygen formation occurring, there is no NADPH formation. There's simply the formation of ATP, and in this process there's only one photosystem one involved. One photosystem involved, it's photosystem 1. Photosystem 2 is absent, and this process occurs in stromal thylakoid, because in stromal thylakoid only photosystem 1 is present, and non-cyclic photophosphorylation occurs in granule thylakoid, because in this there's photosystem 2 and 1 present both. Now in photosystem 1, here photosystem 1 has a characteristic. The photosystem 1 will not produce NADPH2, because here NADPH2 basically requires uh, NADPH2, uh, formation of NADPH2 requires the requires certain types, it has requires an association with photosystem 2, such that if photosystem 2 is present, then there would be a loss of electron, as a result of which, if there is photosystem 2, there would be a loss of electron. With every photolysis of water, there would be a loss of electron, due to which there would be the transfer of electron towards the NADP reductase, due to which it would be able to reduce uh, you know uh, NADP into NADPH2. And moreover, you might be wondering that why NADPH2 is not formed in cyclic phosphorylation, even though photosystem 1 is also present. So reason behind it is that photosystem in cyclic photophosphorylation, NADP reductase is also absent, such that NADP reductase is associated with photolysis of water. So in NADP reductase, since is also present in cyclic photophosphorylation occurs. Now here, as you can see, when light absorbs, light is absorbed by the reaction by the auxiliary pigments, and then the reaction center loses its two electrons. These two electrons are then given towards the ferredoxin, and ferredoxin, instead of transferring it towards the ferredoxin instead of transferring it towards the NADP reductase, transfers it towards the plastoquinone, due to which it becomes plastoquinol, and that it transfers it towards the cytochrome BF complex, and that cytochrome BF complex takes those proton and electron and transfer it towards transfers the proton towards the thylakoid space, and transfers the electron towards the plastocyanine, and plastocyanine now transfers these electron back towards the photosystem 1, uh, as a result of which the electron which was lost by photosystem 1 is regained by its own photosystem. Apart from which, the proton gradient, which ultimately forms here, again cause the process of chemiosmosis, due to which electrochemical gradient is formed, and electron is transferred from the thylakoid is switched to the stroma with the help of CF0, CF1 particles. In this process, CF0, CF1 particles of the ATP synthase complex and ATP synthetase complex, um, or ATPase complex. Moreover, three protons were required to form one ATP. So at least we require a minimum of 54 protons. So in this case, ATP is generated, and this process occurs when there is requirement of ATP as well as due to any other additional process. So this is a basic process about it. And apart from that, there's no involvement of manganese cluster Z complex in it as well as there's no evolution of no utilization of water in this process. Now coming to summary, that finally four important events take place during light dependent radiation: photolysis, electrotransport chain, reduction, and photophosphorylation. Now coming to the next part, which is a independent reaction, Kelvin-Benson cycle, dark reaction, or it is also known as C3 cycle because it occurs in C3 plants. C3 plants are those plants which in which C3 cycle occurs, and C3 cycle is light independent reaction. I mean, it is a Kelvin-Benson cycle in which the first stable product obtained is a 3-carbon sugar which is known as phosphoglycerate, 3-phosphoglycerate. Now it is independent reaction enzymes in the stroma use the chemical energy of the carrier molecules ATP and NADPH2 to drive the synthesis of glucose or other organic molecules. Light independent reaction, dark reaction. In this process, light is not used. This process was explained by Kelvin and Bensel, who were two graduates of Cambridge University and who actually worked on this and won a Nobel Prize for this as well in the form of this process occurring here is in the form of a cycle. The important steps of Kelvin-Benson cycle are as follows: carboxylation, reduction, and regeneration. When it comes to the process of carboxylation, it's quite simple in such a manner that here, first and foremost, the carbon fixation occurs in this process. The carbon dioxide combines with organic molecules, ribulose bisphosphate, and forms a 6-carbon compound called the keto acid. It forms a 6-carbon compound such that ribulose bisphosphate, it is basically, let me just try to explain here. Ribulose bisphosphate is a 5-carbon sugar. Ribulose 5-carbon sugar is there, and that is having bisphosphate, which means it is having two phosphate group at first and fifth carbon, due to which it is known as ribulose bisphosphate. Now ribulose bisphosphate, two ribulose bisphosphate, two ribulose bisphosphate, sorry. So yes, three ribulose bisphosphate over here. Three ribulose bisphosphate will react with three carbon dioxide molecules using the enzyme which is known as Rubisco. Rubisco, it is basically known as ribulose bisphosphate carboxylase oxygenase. It has the capability to perform carboxylation as well as oxygenation. It is more affinity with carbon dioxide, but in case of low carbon dioxide concentration, then it will perform oxygenation. If carbon dioxide and oxygen are present in equal amount, then at that moment it will perform carboxylation because it has more affinity with carbon dioxide. Likewise, Rubisco basically performs a carboxylation such that it fixes the ribulose bisphosphate with carbon dioxide, due to which it forms 6 molecules of 3-phosphoglycerate. And once these steps occur, the next step basically involves the phosphorylation. Or in this process, basically there will be an enzyme utilized transphosphorylase, the process which I initially mentioned in the start of the video, in which 3-phosphoglycerate will be now converted into 1,3-bisphosphoglycerate. There's 1,3-bisphosphoglycerate using the enzyme transphosphorylase. Now, basically, is now further undergone another process in which which would be known as 1,3-bisphosphoglycerate. In this there would be reduction. Reduction would be in such a manner that here, a 3-phosphoglycerate over here, 1,3-bisphosphoglycerate. I'm just writing it for example. 1,3-bisphosphoglycerate over here will be now converted into uh, 3-phosphoglyceraldehyde, 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 such that this 3-phosphoglyceraldehyde will be formed, and that 3-phosphoglyceraldehyde will be formed with enzyme 1,3 bis phosphoglycerate reductase, and that 3-phosphoglyceraldehyde is since there are, how many molecules, since we have got 12 molecules of 3-phosphoglycerate, there will be formation of, sorry, 6 molecules of 3-phosphoglycerate, there will be molecules of 6 molecules of uh, 3-phosphoglyceraldehyde. After that, these 6 molecules of 3-phosphoglyceraldehyde will be obtained, apart from which, you know one molecule one molecule of 3-phosphoglyceraldehyde will uh, be taken outside, while 5 molecules of 3-phosphoglyceraldehyde will be now, regenerated using the enzyme ribulose bisphosphate, and converted back into the ribulose bisphosphate. Here, the process which would be occurring would be basically, using the enzyme. Here, occurring would be the enzyme which would be utilized in this process would be, a reductase. Here, glyceraldehyde 3-phosphate basically now will be, oxidized, as a result of which it will cause to be converted into uh, 3 molecules of ribulose bisphosphate. And here, this is how the process of photosynthesis will be occurring over here. Apart from the theory is also written over here, such that here in the reduction process, this is the following reaction occurring: 6,3 phosphoglyceraldehyde plus 6 NADPH plus 6 ATP forms 6 molecules of 3 phosphoglyceraldehyde plus 6 NADP and 6 ATP and 6 inorganic phosphate. This is, which means 6 NADP are, consumed in this process. 6 ATP are also consumed in this process. Whereas in the process of forming 3-phosphoglycerate, there's neither there's only consumption of, there's no consumption of NADP and ATP, but there is a consumption of ATP, I guess. There's no consumption of ATP. So here there will be 6 ATP and 6 NADPH2 are already consumed. Now in this case, the regeneration process, in this process carbon accepted ribulose bisphosphate is consumed and is formed again, and its regeneration takes place. 3 carbon compounds are arranged to form 5 carbon unit, such that here 5 3-phosphoglyceraldehyde reacts with 3 ATP to form 3 ribulose bisphosphate, 3 ADP plus 2 inorganic phosphate, such that now we have utilized 3 ATP here, 6 ATP previously, 9 ATP in one uh, Calvin cycle, and this Calvin cycle occurs 2 times, as a result of which 2 molecules will be obtained. Uh, so 2 ATP molecules, I mean 2 times, the cycle will occur 2 times basically. So as a result, because we want hexose, one turn gives us triose, so 2 turns will give us 2 triose, which will be further converted into hexose. Now here 6 ATP were obtained in the reduction step, 3 ATP were obtained in the regeneration step, 9 ATP were obtained in a single cycle. So in two cycles, there would be 18 ATP. Likewise, in one cycle, there are 6 NADPH consumed during the reduction step. So in 2 cycles, there would be 12 NADPH to consume. Likewise, in one cycle, there is, 9 ADP utilized, and in one cycle there is 9 ADP utilized, in 2 cycles 18 ADP utilized. In one cycle there is 8 ADP, 8 inorganic phosphate released, and in 2 cycles there will be 16 8 inorganic phosphate released. Now you might be wondering that why there are 2 inorganic phosphate less than the number of ATP. Reason behind it is that the 2 inorganic phosphate will now be released further during the formation of hexose from the triose, which is released, known as glyceraldehyde 3-phosphate, because it is also having a phosphate group. So 2 cycles having 2 glyceraldehyde 3-phosphate will tend to, release 2 of the phosphate group, as a result of which it will cause to form hexose sugar, and 2 phosphate group will be obtained from there, so that they can be utilized in the process of cyclic or non-cyclic photophosphorylation or in the light reaction. This compound is used to start the cycle over again, and this is how the reaction basically occurs. After that, the topic which we are going to discuss is the process which is known as the photorespiration. We are skipping this part, coming to the next part in which we are going to discuss about the photorespiration in plants. Photorespiration here is an important topic because here it involves, conversion. Uh, First and foremost, it is basically a wasteful process. It is, neither similar to photosynthesis nor it is a complete respiration. Neither it is a complete respiration nor it is a complete photosynthesis. So it is intermediate between both of them, and it is a wasteful product process. In such a manner that here, as you can see, sugar is oxidizing the chloroplast during daytime product without production of energy or ATP, which is called photorespiration. In this process, carbon dioxide is released and oxygen is absorbed as seen in aerobic respiration. In other words, we can say that Rubisco enzyme, instead of performing carboxylation, performs oxygenation, and oxygen combines with a compound which is called ribulose bisphosphate. In this process, an enzyme Rubisco is utilized, and that causes to produce two components of RUBP, such that instead of producing phosphoglycerate, it causes to produce two compounds. First includes phosphoglycerate, three carbon sugar, and phosphoglycolate. Now, phosphoglycerate, which is obtained, will go inside the Kelvin Benson cycle. But problem would be there that the quality of the sugar obtained from the phosphoglycerate obtained through photorespiration will not be as effective as good nutritious as compared to quality of that which is obtained through the process of the pure Kelvin Benson cycle. So in this case, this would be a condition after that phosphoglycolate here will undergo the photorespiration process. Let's see how it actually happens. Now, glycolate basically produced during the process diffuses into the membrane bound organelle known as peroxisome, and uh, first and foremost, this phosphoglycolate here, dephosphorylation of phosphoglycolate occurs, as a result of which phosphoglycolate is converted into glycolate, as a result of which this phosphoglycolate now will be sent into the peroxisome, which is a membrane bound organelle. And here in peroxisome there is an enzyme known as glycolic acid oxidase, as a result of which it will cause oxidation of glycolate and convert into uh, uh, glyc uh, you know uh, from glycolate it would be converted into glycine. And it would be not that simple, because here glycolic acid oxidase will cause to convert glycolate into glycine, and then glycine, basically it is an amino acid, and glycine rapidly diffuses into the mitochondria, where two glycine molecules convert into a single amino acid, which is known as serine, which is a three carbon amino acid, and one carbon dioxide is evolved. That carbon dioxide is again evolved in mitochondria, which is of no use in mitochondria, while in serine there is three carbon compound, which is a three carbon amino acid, which is known as serine. And apart from that, um, this process is done by the help of an enzyme which is known as glutamate transaminase. In peroxisome, there is it is a chain of reaction. So in peroxisome, the reaction which would be occurring here would be first and foremost, glycolic acid oxidase will act, due to which it will convert the glycolate into glycolate into another sugar which is known as glycerine, and from glycerine it reacts with glutamic acid in order to form, using an enzyme, glutamate transaminase, due to which it causes to produce glycine. So this is a chain of reaction occurring over here. Apart from that, that glycine obtained will be now converted into serine and carbon dioxide in the mitochondria. Now within that process, that serine will be, carbon dioxide will remain within mitochondria, and serine will be now released, sent to the peroxisome, where it would be converted into hydroxypyruvate, from where it would be converted into pyruvate, and from the pyruvate it would be converted back into, uh, it would be sent into the chloroplast, where it would be converted from the pyruvate into uh, glycerate, from where it would be converted from the pyruvate into glycerate, 3-phosphoglycolate, and from 3-phosphoglycolate, it would be converted into 3-phosphoglycerate. So in this case, this is how the reaction will occur. By the way, it's not glycolate, it's glycerate. So first and foremost, when pyruvate will be formed, pyruvate will be converted into glycerate, and glycerate will further be phosphorylated to form 3-phosphoglycerate. So this is the reaction which would be occurring. Now coming to its disadvantages, because it's of no use. This process basically decreases the net potential of photosynthesis by 5%, as well as has a very harmful effect that it decreases the 25, it decomposes 25% of the ribose bisphosphate present within the cell, which is supposed to be utilized for the formation of hexose. And photorespiration is wasteful and is without any benefit process. In this process, energy is not evolved. ATP and NADPH2 are not synthesized. On the contrary, both are produced in the right. Those which are produced in the right direction are also consumed, as well as carbon dioxide is released. The growth of plant is also reduced up to 25% due to photorespiration. It also reduces photosynthesis, as a result of which crop production may be reduced. So these are some advantages. I mean, these are some disadvantages of it, which that is why there are some alternative mechanisms in order to withdraw it. Dry and hot weather, the stomata of leaves are closed due to the to reduce the rate of transpiration, and this occurs through the abscisic acid hormone, which causes to decrease close the leaves of close the stomata, due to which it reduces the rate of transpiration. So there is less supply of carbon dioxide, and amount of oxygen inside the plant is increased, because there's ongoing process of uh, there's ongoing process occurring there is an ongoing process of light reaction, due to which oxygen is evolved, but carbon dioxide is not being entered, due to which because gaseous exchange is not is occurring in this process. Plants consume oxygen and produce carbon dioxide during daytime in the chloroplast without production of energy, so it is termed as photorespiration. To avoid photorespiration, there are two alternative mechanisms over here, which include C4 cycle and CAM pathway. First, including C4 cycle over here. C4 cycle is arranged in such a manner. C4 cycle, it is also known as Hatch and Slack pathway. H. and Slack was the name of the scientist, and C4 cycle occurs in particularly C4 plants, those plants which live in dry and tropical uh, places, as well as C4 plant include examples of Z maize, Saccharum officinae, sugarcane, as well as Borgum, Sorghum bicolor are example of this, and it is also known as dicarboxylic acid cycle. The reason behind it is that first and foremost, it is having its first stable compound formed as a 4 carbon containing sugar, which is known as oxaloacetate, and that oxaloacetate has got two carboxyl group within its structure, due to which two carboxylic acid group within its structure, due to which it is known as dicarboxylic acid cycle or C4 cycle. In some plants, another metabolic cycle is used to fix carbon dioxide in the presence of high concentration of oxygen, which is known as C4 plant. They develop anatomical and physiological modification, develop some tissues around the vascular bundle called bundle sheath, which is known as Crane's anatomy. Cranes means wreath, which means arrangement in the form of ring, such that they have a bundle sheath around which they have cells known as bundle sheath cells in a circular arrangement, and around the circular arrangement there is presence of certain types of cells which are known as mesophyll cells. Let me just try to draw a structure over here. Let's suppose this is the adaxial epidermis, apexial epidermis. Just trying to draw a rough structure, and here in between there is a vascular bundle having xylem phloem, vascular bundle, and around the vascular bundle there is arrangement of there's a a membranous sheath over here, membranous sheath. There is arrangement of a membranous sheath, which is known as the bundle sheath, and around this membranous sheath there are cells which are known as bundle sheath cells. Bundle sheath cells. Now these are the bundle sheath cells, and around these bundle sheath cells we are having uh, presence of some cells. First and foremost, bundle sheath cells are in circular arrangement, and around this bundle sheath cells There are certain cells in circular arrangement known as mesophyll cells. Certain cells in circular arrangement, mesophyll cells. These are the mesophyll cells which are arranged in circular arrangement. As you can see, these are the bundle sheath cells around which there is the mesophyll cells in circular arrangement. And instead of instead of the process of Kelvin Benson cycle in mesophyll cells, there is another process occurring in the mesophyll cells which is known as the process of C4 cycle. C4 cycle will be occurring in the mesophyll cells, and in bundle sheath cells there will be process of Kelvin Benson cycle. Now first let us discuss about some features of the Crane's anatomy, such that Crane's anatomy says that here that this is the structure in which the structure is obtained. I know it's a bit um, you know uh, a bit messy, but you can better acknowledge it. Now here what we are going to do is to study the structure. Here the C4 plants are having dimorphic chloroplasts, and usually plants we are having a monomorphic chloroplasts, but here we are having dimorphic chloroplasts, such that in bundle sheath cells we are having bundle sheath cells we are having large a gran a granular chloroplast, if I'm not wrong, a granular chloroplast, which are less in number. Likewise, in the mesophyll cells, there is small granule chloroplast. These are not granular, granule chloroplast. They do not have granum. Uh, Small granule chloroplast, granule chloroplast, which are more in number. Right. After that, when it comes to second part, here bundle sheath cells are basically having uh, the process of Kelvin cycle, C3 cycle, and here there is C4 cycle. So for that, there is enzyme pepcase, phosphoenol pyruvate carboxylase, and here there is enzyme Rubisco. Pepcase is a specialty that it does not perform oxygenation but only performs carboxylation. Likewise, in order and bundle sheath cells are basically having thick, sorry, bundle sheath cells. Oops, there would be there's a mistake that here there is presence of Rubisco. Kelvin cycle occurs in bundle sheath cells. So here there is presence of Rubisco. Here there is pepcase, right? And here in the bundle sheath cells, um, they are having permeable thick permeable cell wall thick permeable cell wall so that thick impermeable cell wall, so that no gases can be permeated. And mesophyll cells have thin permeable permeable cell wall, thin permeable cell wall, right? After that, when it comes to some other part, uh, which is a basic difference, since they are agranular. They are granular. There is no granum present. There will be no process of photolysis occurring, or in general, there will be no light reaction. Light reaction, no, and light reaction, yes, and photolysis, no, photo photolysis, yes, oxygen evolution, no, and oxygen evolution, yes, oxygen evolution, yes, right. So this is a basic, and after that, when it comes to some other part, um, here this is a basic difference between Crane's anatomy. And when it comes to structure, let us discuss it over here. Here, as you can see, the cycle occurs in such a manner. Let me just explain with the diagrammatic manner. Here, as you can see, there is first and foremost the C4 cycle occurring. C4 cycle basically occurs in such a manner that in C4 cycle, the phosphoenol pyruvate causes the phosphoenol pyruvate causes to react with carbon dioxide, which is obtained from the process of respiration. So through the process of respiration, the carbon dioxide which is obtained causes to combine with phosphoenol pyruvate, which means carbon dioxide fixation occurs over here through the enzyme which is known as peptidase, as a result of which a C4 acid is formed. This C4 acid which is formed over here is basically known as oxaloacetate, which is a first stable compound, 4 carbon compound for oxaloacetate. The C4 acid further, now as this oxaloacetate is formed, that oxaloacetate further now goes into the oxaloacetate now further goes into the bundle sheath cells, and within the bundle sheath cells the oxaloacetate causes to become decarboxylated. As a result of decarboxylation, it releases carbon dioxide and forms a C3 acid. This C3 acid is basically pyruvate, and that pyruvate again is transported back into the mesophyll cells, from where it is transported to from where it is phosphorylated by the addition of phosphate, due to which it causes to form phosphoenol pyruvate on one hand side, and on the other hand side the phosphoenol pyruvate on one hand side, and on the other hand side carbon dioxide release, which is utilized in the process of Kelvin Bunsen cycle. Now this is how the basic mechanism occurs, and apart from that, when it comes to the cam cycle is also simple in such a manner that here it is an adaptation occurring in the plants of crassulaceae family. Crassulaceae family, um, examples of this includes euphorbia, birophyllum, and agave. Moreover, other succulent plants or xerophytic plants which survive in dry and tropical condition, which includes cacti, pineapple, and many and rang. These are some plants crassulacean in which there is crassulacean acid metabolism cycle occurring. Acid refers to the word malate because the metabolism will be occurring of an acid which would be a 4 carbon acid malate, right? So here this would be the acid of which the metabolism will be occurring. After that, uh, this they basically have a characteristic that they have scotoactive stomata instead of photoactive stomata, such that they have stomata which open during the night and close during the daytime, which are known as scotoactive stomata. So for that, the adaptation is in such a manner that here their mode of carbon dioxide fixation is called crassulation acid metabolism, or CAM. The plants are known as CAM plants, CAMP, and CAM plants store these organic acid with carbon dioxide in their vacuoles. And now let us study how. So basically, during night time, there are two processes occurring: primary carboxylation and secondary carboxylation. So primary carboxylation occurs during the night time, and secondary carboxylation occurs during the daytime. Primary carboxylation has the primary carbon acceptor as phosphoenol pyruvate, and secondary carbon carboxylation has its secondary carbon acceptor at as ribulose bisphosphate. Let's study how. So during night time, this is a night time when stomata are open, due to which carbon dioxide is being taken in. So when carbon dioxide is taken in, the peptidase enzyme basically causes to fixate carbon dioxide into phosphoenol pyruvate, due to which it causes to form oxaloacetate. Now oxaloacetate is further converted into malate using the enzyme malate dehydrogenase, in which there is requirement of one NADH molecule. And once malate is formed, that malate is then released into stored into a vacuole which is known as malic acid vacuole. And this is the process which occurs during the uh, this is a process which occurs during the night time. And during the daytime, what actually happens is that the stomata are closed. As a result of closure of stomata, it causes the malate which was formed to come out, And as a process of malate will occur, malate will come out. Malate basically will now be decarboxylated with the enzyme NAD plus malk enzyme, which will cause to decarboxylate it, and pyruvate will be formed on one hand side, and carbon dioxide will be formed on another side, which should be released as Calvin cycle, and pyruvate will be now sent towards the chloroplast, where it would be stored in the form of starch. Pyruvate will be now further at that moment will be phosphorylated and converted into phosphoenol pyruvate. This is the basic process occurring. And apart from that, the next step we are going to study is about the air respiration. Here, as we can see, that now we are going to study about cellular respiration. Respiration is an exchange of gases. In this process, oxygen enters the body and carbon dioxide is given out. Now, cellular respiration, basically it is the process in which there is oxidation of glucose such that aerobic breakdown of molecules occurs with the formation of ATP, which is known as cellular respiration. It can be of two types. First includes anaerobic respiration, fermentation. It is a term for anaerobic respiration, and alternative term for anaerobic respiration, fermentation. Energy respiration takes place without oxygen. It was defined by Pasteur, Louis Pasteur. It is of two types of alcoholic fermentation and ethyl alcohol, where ethyl alcohol is produced from pyruvate, and lactic acid fermentation when lactic acid is produced from pyruvate. This process less amount of energy is released, around 2 ATP. Net ATP releases 2 ATP. Moreover, when it comes to other ATP, some organisms, especially microorganisms, obtain energy by anaerobic respiration, such as yeast, Saccharomyces cerevisiae, and some bacteria, such as Lactobacillus acidophilus. In both kinds of respiration, the first phase is glycolysis, in which oxygen is not required and glucose, after several reaction, converts into two molecules of pyruvate or pyruvic acid. We're going to discuss that in detail. Here, the process of glycolysis is divided into two parts. Initial stage of respiration is called glycolysis, in which oxygen is not used. It is common or universal phase. In aerobic and anaerobic respiration, and glucose molecule is broken down into two molecules of pyruvate. The different steps are governed by enzymes which are present in cytosol. Along with enzymes, ATP and NAD are also required. It can be divided into two phases. As you can see, first is preparatory phase. Preparatory phase is the one in which preparatory phase, or in other words, we can also call it as investment phase, in which glucose is converted to the phosphorylated triose or glyceraldehyde 3 phosphate. How? Let's see in the diagram. First step basically involves phosphorylation or glucophosphorylation, in which there's addition of phosphate on the glucose 6 carbon, as a result of which it forms glucose 6 phosphate, and ATP molecules is invested in it, due to which the enzyme utilized over here is phosphohexokinase or glucokinase. After that, second step involves uh, isomerization. Isomerization basically is done by an enzyme which is known as phosphohexoisomerase, due to which it is converted into a fructose 6 phosphate. After that, fructose 6 phosphate is then converted into fructose 1,6 bisphosphate or fructose 1,6 diphosphate with the help of an enzyme. This process is known as phosphorylation or fructophosphorylation, in which there is utilization of an enzyme which is known as phosphofructokinase. Moreover, when it is done, fructose 1,6 diphosphate is now dissociated into two molecules. Splitting occurs of fructose 1,6 diphosphate into one molecule of glyceraldehyde 3 phosphate and another molecule of 3 dihydroxyacetone with the help of an enzyme which is known as aldolase. Aldolase is having its active site made up of glycine, alanine, and histidine amino acids, due to which it causes splitting and it causes to produce the two molecules 3 glyceraldehyde 3 phosphate and 3 dihydroxyacetone. Now here 3 dihydroxyacetone uh, now is further, this is one step is not written over here, which is of isomerization, such that 3 dihydroxyacetone is now isomerized using the enzyme phosphotriose isomerase into the glyceraldehyde 3 phosphate, as a result of which two molecules of glyceraldehyde 3 phosphate are involved. Now the next step, which this is till here it is completed, or preparatory phase or investment phase, in which two molecules of ATP are invested, no molecule of NADH is formed, as well as uh, this is a reaction till now, and we have obtained glyceraldehyde 3 phosphate, two molecules. So now coming to the next part involves the preparatory phase or payoff phase. In this phase, the first step involves the dehydrogenation, and um, first step basically involves dephosphorylation and dehydrogenation. sorry, dehydrogenation and phosphorylation, sorry. Dehydrogenation and phosphorylation. Dehydrogenation first and foremost occurs such a manner. First and foremost, there will be phosphorylation. Phosphorylation will occur of glyceraldehyde. Glyceraldehyde will be converted into 1,3-glyceraldehyde phosphate, and this will be a non-enzymatic reaction, which is usually not written in books. And then the glycerol 1 dehyde 1,3 phosphate will now be converted into 1,3 uh, phosphoglycerate, 1,3 phosphoglycerate, which means dehydrogenation will occur using an enzyme which is known as phosphotriose dehydrogenase, and triose phosphate dehydrogenase. So this is the enzyme utilized over here. These both are the coupled reaction, which means it occurs together. Uh, After that, the two glyceraldehyde 3 phosphate are converted into diphosphoglycerate. Now diphosphoglycerate here, uh, this is the first step in which dehydrogenation and phosphorylation occur together. After that, a diphosphoglycerate will now be converted into phosphoglycerate, releasing two molecules of ATP. One more thing, that this preparatory phase occurs two times, which means as two molecules of glyceraldehyde 3 phosphate are formed, which means it will occur two times separately in two chains. Now diphosphoglycerate is formed, which basically now the step which will occur over here will be the formation of ATP. Formation of ATP will occur by an enzyme which is known as phosphoglycerol transphosphorylase or phosphotransferase. So this would be the enzyme utilized. Two ATP molecules will be formed, and now the condition would be phosphoglycerate. Now, 3-phosphoglycerate now will be isomerized into 2-phosphoglycerate with the help of an enzyme which is known as phosphoglyceromutase. After which after which 2-phosphoglycerate will be formed. 2-phosphoglycerate will now will further, you know, there will be hydration occurring in it. Hydration will lead to the this process, hydration will lead to the formation of phosphoenol pyruvate with the help of an enzyme known as enolase. And after that, if phosphoenol pyruvate will now have formation of ATP, such that there will be enzyme used pyruvate kinase, which will cause formation of ATP by dephosphorylation of phosphoenol pyruvate, due to which two molecules of pyruvate will be formed, and two molecules of ATP will be formed. And in that case, we have 4 ATP gained. 4 ATP are the amount, gross ATP. Now, in order to gain the net ATP, it will be around 2 ATP molecules. Now, 2 pyruvate, basically, in case of lactic acid fermentation, uh, the 2 pyruvate will be converted into 2 molecules of lactate using the 2 NADH molecules which are prepared over here using the enzyme lactate dehydrogenase. In case of in case of alcoholic fermentation, ethanol fermentation, first pyruvate will be converted into acetaldehyde with the help of an enzyme known as pyruvate decarboxylase multi-enzyme complex, after which the acetaldehyde, also there will be release of 2 carbon dioxide, and then acetaldehyde will now be further converted into ethanol through the process of reduction occurring in it. Now, in that case, the enzyme utilized will be ethanol dehydrogenase or alcohol dehydrogenase, due to which the process will occur. Now, reduction will also accompany the use of two NADH molecules which are produced in the glycolysis in case of alcoholic fermentation, and same, there will be two carbon dioxide, two NAD plus ethanol, as well as two ATP molecules which are net gain. Now, this is a theory. After that, when it comes to aerobic respiration first includes oxidation of pyruvic acid. This is a gateway step between glycolysis and Krebs cycle. It is also known as acetylation of pyruvate, such that pyruvate does not enter directly. uh, Basically, when pyruvate enters into the mitochondria, it requires to enter by the help of a transport protein known as transcolase, through active transport, so that it enters inside. And when it is entered inside, it basically converts into it is converted into acetyl CoA by the process of oxidative decarboxylation, such that first and foremost the structure will be there will be decarboxylation occurring using the same enzyme, pyruvate decarboxylase. Then pyruvate decarboxylase will over here, as a result of which pyruvate decarbooxylase, a multi-enzyme complex, will cause to form acetaldehyde plus 2 carbon dioxide. Now acetaldehyde will further be oxidized through pyruvate dehydrogenase, multi-enzyme complex, due to which there will be formation of acetyl plus two molecules, acetyl, and in that case acetyl will be formed, and that acetyl now will be attached with coenzyme A. Here we are going to discuss its composition. Coenzyme A is in such a manner: if we are having adenosine having if adenosine is having a phosphate group at its third carbon as well as at its fifth carbon, and it is combined with a molecule of vitamin B5, which is pantothenic acid. It is basically having four phosphate group attached to it. So four phosphate group attached to the pantothenic acid will be named as 4-phosphopantothenic acid, and that 4-phosphopantothenic acid, when will attach with the fifth carbon of the adenosine 3,5 bisphosphate, it will cause to form a structure which would be known as coenzyme A. And that coenzyme A will be utilized over here. Coenzyme A alongside acetyl combine together, due to which it will form acetyl CoA. Right. After that, when it comes to other part, there is Krebs cycle. First acetyl CoA is formed. It is then taken inside the cell through the active transport through the mitochondria, through the active transport by the transport protein transcolase. After that, Krebs cycle, acetyl CoA links glycolysis with Krebs cycle. It faces its acetyl group into the Krebs cycle for further oxidation, and here the process occurs in such a manner. The first step into synthesis occurs in such a manner: acetyl CoA is converted into citrate. It is also known as citric acid cycle or tricarboxylic acid cycle, because citrate contains three carboxylic group. And here the enzyme utilizes citrate synthetase, due to which citrate is formed. After that, citrate, when it goes through a coupled reaction, in which a coupled reaction of dehydration and rehydration, in which citrate gets dehydrated with the help of enzyme aconitate, due to which it forms 6-aconitate, after which 6-aconitate further gets rehydrated with the help of the same enzyme aconitate, because it is a coupled reaction, so same reaction will be occurred by the same enzyme. And now 6-aconitate will be now rehydrated into isocitrate, and now once it is done, isocitrate will now be further, there will be another coupled reaction, which would be oxidative decarboxylation, such that oxidation first will occur. Oxidation will lead isocitrate dehydrogenase will be first converted into it will be oxidized into oxalosuccinate using isocitrate dehydrogenase, and then oxalosuccinate will be now further decarboxylated using the same enzyme isocitrate decarboxylase into alpha ketoglutarate, which is basically a sugar which is obtained due to deamination of glutamic acid. Now, alpha ketoglutarate is a 5-carbon sugar, which is now further, undergoes a coupled reaction, oxidative decarboxylation, in which the enzyme alpha ketoglutarate decarboxylase is utilized. Sorry, alpha ketoglutarate dehydrogenase is utilized, in which first step includes, dehydrogenation, and then includes decarboxylation, as a result of which it causes to form a product obtained, which is known as succi which the product obtained, which is known as succinyl. Let me see here. As you can see, it causes to form a structure which is known as succinyl. Now succinyl basically is due to the oxidative decarboxylation of alpha ketoglutarate. As a result of which, succinyl is a 4-carbon sugar, and now succinyl will now further combine with CoA with the help of an enzyme which is known as CoA. Succinyl CoA, due to which it will combine due to an enzyme which is known as succinyl CoA succinyl CoA synthetase. After the succinyl CoA synthetase, when it will be formed, succinyl CoA synthetase will now be further, succinyl CoA synthetase, which is now further formed, will cause to, be converted into succinate using the enzyme succinate thiokinase. Succinate thiokinase basically will cause to take the inorganic phosphate from the succinyl CoA, as a result of which that inorganic phosphate will be transferred to the GDP, due to which it will be converted into GTP as a result. on one hand side GTP, which is formed, is energy rich, due to which it will react with ADP. GTP reacting with ADP under the action of an enzyme which is known as nucleoside diphosphate kinase will cause to yield ATP with GDP. This process is known as substrate level phosphorylation, which will occur over here. After that, on the other hand side, succinyl CoA will be converted into succinate, and on the other hand side, the succinate which is formed will now further be dehydrogenized with the help of an enzyme which is known as succinate dehydrogenase, which causes to convert it into fumarate. Now here succinate dehydrogenase has a dual role in Krebs cycle as well as electron transport chain. In electron transport, it is basically located in the inner membrane of Krebs cycle, such that inner membrane of Krebs cycle, it is located in complex number 2, which is known as FADH dehydrogenase complex, in which it is located, where it causes the dehydrogenation of FADH into FAD, as well as it causes the dehydrogenation of succinate into fumarate. Moreover, fumarate now will further be hydrated. Moreover, at this stage, when succinate will be converted into fumarate, there will be production of FAD into FADH, such that FAD will be reduced into FADH. Likewise, when it comes to fumarate will now be hydrated, such that hydration of fumarate will occur through an enzyme which is known as fumarase, as a result of which fumarase will cause it to convert into malic acid, and now malate basically will now be converted into again oxalosuccinate, using the same pathway from where the cycle started, uh, through the enzyme malate dehydrogenase. And this is the entire process explained within a few explained here. And yes, this is the entire process. And now after that electron transport chain, it is the oxidative phosphorylation. Electron transport chain is the last step here in the process of glycolysis. Krebs cycle, hydrogen is released, which is transferred to different compounds like NAD, NADP, and FAD. These are called hydrogen acceptor carriers. Now the effect of hydrogen is transferred from one compound to another to produce energy rich compounds, ATP. This type of phosphorylation is oxidative phosphorylation. Now in this case, there are several complexes which are present on the inner membrane of mitochondria, which are, among which we are going to study four main complexes. Co you know First complex includes, first complex basically includes complex number 1, NADH dehydrogenase complex, after which there is FADH dehydrogenase complex. Second complex, third complex is cytochrome reductase complex, after which there is cytochrome oxidase complex, complex number 3 and 4 respectively. And complex is also there, which is known as ATP synthetase complex. But we are going to only study the four. Then, of course, it is a different part. It will include chemiosmosis, which we are going to study later. After that, when it comes to NADH dehydrogenase complex, basically what actually happens is that, um, um, when NADH is entered within that NADH dehydrogenase complex, the NADH dehydrogenase complex causes to take the two electrons as well as two hydro protons from NAD, and causes to pump the proton from the NADH from causes to pump the proton from the matrix to the lumen, as a result of which protons are pumped into the intermembranous space, while electrons are transported towards a coenzyme which is known as coenzyme Q or ubiquinone. And that ubiquinone basically transfers the electron from the complex number 1, complex number 2, to the complex number 3, which is cytochrome reductase complex, right? And cytochrome reductase complex. And after that, uh, in this case the electrons are transported to cytochrome complex number 3, and protons are pumped only when there is movement of electron. When there is movement of electron will be move electron will be accepted, then at that moment there will be pumping of proton. Due to the energy of electron, the protons will be pumped from the matrix to the intermembranous space. As a result, as you can see, we are having a pumping of protons. Pumping of proton occurs in complex number 1, 3, and 4, but not in complex number 2, because complex number 2 doesn't has any sufficient energy to pump protons. As a result of which, due to the movement, due to no sufficient energy, FADH dehydrogenase complex does not pump proton. And when it comes to movement of electron, one electron movement of one electron will generate one proton from the matrix to the intermembranous space. So one proton by NADH, one proton generated by cytochrome reductase, one proton generated by oxidase. As a result of which, three protons will be pumped due to uh, three proton will be pumped due to complex number 1, 3, and 4, due to which there will be due to which we can say that NADH will produce three proton, while on the other side FADH, since FADH dehydrogenase complex will not pump proton, the like protons here the number of protons pumped would be zero, while protons will be pumped on cytochrome reductase complex and cytochrome oxidase complex, due to which only two proton will be pumped in case of FADH. So FADH will have two number of protons pumped. Two number of protons will be pumped in the case of FADH. So this is how the two number of protons will be pumped in the case of FADH, and this is the reason why it occurs. And here on the other hand side, one more thing, that here uh, the cytochrome reductase complex is further composed of cytochrome B1, cytochrome B2, cytochrome B naught, cytochrome B naught, cytochrome B1, and cytochrome B1, and cytochrome C1 complex. Now when cytochrome reductase complex, this completely forms a cytochrome reductase complex, when donates its electron towards the cytochrome oxidase complex. The cytochrome oxidase complex basically it does not directly do that. Again, there is another carrier which is on a cytochrome C. Cytochrome C carries that electron and then transports it towards the cytochrome oxidase complex. And here uh, this is how the transport occurs. Now during the transport, the number of protons which are generated over here, number of protons which are generated over in the intermembranous space will cause the protons generated in the intermembranous space, the protons which are generated in the intermembranous space will cause to create an electrochemical gradient, due to which again chemiosmosis will occur, due to which the protomotive force will be generated, and thus the ATP synthetase enzyme will pump the proton from the lumen to matrix using the protomotive force, and thus due to the due to the concentration, there will be concentration gradient, there will be protomotive force generated, due to which it will be able to pump the proton from the lumen to the matrix, as well as ATP synthetase enzyme will be activated and cause ATP synthesis, or we can also call this as F1 particle, will cause to convert ADP into ATP on one hand side, and on the other hand side, uh, when it comes on the other hand side, on the other hand side, the F naught particle basically is a transmembrane protein which helps in pumping of protons from the lumen to the matrix. And apart from that, these are some steps written. And now coming to cellular respiration of proteins and fats. Cellular respiration of proteins and fats is in such a manner that if we are having amino acids, proteins are dissociated into amino acids. Amino acids will have two parts. Let me just try to draw a structure. It is asymmetrical carbon, radical group, hydrogen, and on the other hand side there is carboxylic group, hydrogen, and here there is amine group. Now here the amine part, this part will be taken during deamination process, in which amino acid will be converted into ammonia. Here ammonia will be formed on one hand side, while the rest of the structure will either go in the pyruvate part of glycolysis, or it will become acetyl CoA and undergo Krebs cycle. Now amino which is formed over here will now further form urea through the urea cycle, or ornithine arginine cycle, urea cycle. And once urea is formed, that urea will further be excreted in the form of urine through the body, right? So this is how the cellular respiration of proteins occur. When it comes to cellular respiration of fats, it occurs in such a manner that here glycerol which is formed, glycerol it will be oxidized, as a result of which it will cause to produce glycerol aldehyde. It is alcohol, so it will be oxidized and form glycerol aldehyde, and since it will form glyceraldehyde, then it will be phosphorylated, due to which it will form glyceraldehyde 3-phosphate, and again it will undergo glycolysis. When it comes to fatty acids will undergo oxidation, due to which they cause to form acetyl CoA and undergo Krebs cycle. This is the basic mechanism of the entire topic, and hopefully we'll meet again in another